This video will cover cancer and the process of oncogenesis, distinguishing between benign and malignant tumors, and three major types of skin cancer. Cancer is a disease that results from uncontrolled cell division. Cell division is usually under strict control to ensure appropriate proportion. When cell cycle regulation fails, cells start dividing uncontrollably and result in abnormal masses of dividing cells called tumors. Oncogenesis, also known as carcinogenesis, is the transformation of healthy cells into cancerous cells. This involves genetic mutations that disrupt the processes which normally control tissue growth through the balance of cell division and apoptosis, or programmed cell death. Tumor suppressor genes code for proteins that normally work to slow down the cell cycle, stimulate DNA repair, or stimulate apoptosis of unhealthy cells. A mutation that disrupts the function of these tumor suppressor genes can prevent the repair of DNA damage and prevent apoptosis, allowing unhealthy cells to continue dividing out of control. The illustration here shows the example of P53, a tumor suppressor gene that normally responds to DNA damage by stopping the cell cycle and stimulating DNA repair, or apoptosis, if the DNA damage cannot be repaired. Mutations that decrease the normal activity of P53 are commonly found in skin cancers. Oncogenes are mutated genes that speed up the cell cycle, contributing to cancer. Proto-oncogenes code for normal cell signaling proteins that stimulate growth. Here we see the example of epidermal growth factor and the intracellular signaling cascade stimulated by the epidermal growth factor receptor. This involves the mitogen activated protein kinase signaling cascade, where RAS kinase phosphorylates and activates MEK kinase, which phosphorylates and activates ERK kinase. ERK then stimulates cell division by activating a variety of different targets that turn on genes within the nucleus to speed up the cell cycle. With the normal functioning epidermal growth factor receptor and signaling cascade, if there's no epidermal growth factor present to stimulate the receptor, the cell signaling cascade is not active. But a mutation in the gene for RAF is commonly found in skin cancers, and mutations associated with skin cancers often lead to increased activity of RAF, so that RAF can stimulate MEK and ERK without epidermal growth factor binding to the epidermal growth factor receptor. This leads to uncontrolled cell division. Benign tumors are masses of abnormal tissue that do not spread to invade other tissues. Acrocordons, or skin tags, are examples. An acrocordon is a fibrous mass covered with the typical epidermal tissue. These often form in skin creases or folds where the skin rubs with movement. A lipoma is a benign tumor where adipocytes grow and form a mass surrounded by a thin, fibrous capsule. A melanocytic nevus, or mole, is a mass of melanocytes that tends to remain relatively stable in size, shape, and color over time. A nevus is a fairly common and benign type of tumor. Normally, it has a symmetrical shape with smooth borders and uniform color, and the diameter is smaller than 6 millimeters. Malignant tumors are cancerous tumors that invade adjacent tissues. Malignant tumors are more dangerous than benign tumors because they have the potential to metastasize, where cells break free from the primary tumor and travel to other areas of the body where they can disrupt the function of organs distant from the initial tumor. Basal cell carcinoma is a form of skin cancer resulting from mutation and overgrowth of the mitotically active stem cells in stratum basale of the epidermis. While this is the most common form of skin cancer, it is the least dangerous because basal cell carcinoma is unlikely to metastasize, spreading to other regions of the body. These cancers are frequently found on the head, neck, arms, and back, areas that are often exposed to sun. Although UV radiation is the main culprit, wounds and exposure to mutagenic chemicals can also increase risk for basal cell carcinoma. The most common form of treatment involves surgery to remove the cancer. Freezing, called cryosurgery, is another option. And there are also topical chemotherapy ointments that can be used. Here we see the histology of a basal cell carcinoma. We can see the abnormal malignant tumor cells are spreading through the dermis. Squamous cell carcinoma is another form of skin cancer. Squamous cell carcinoma forms from uncontrolled growth of the keratinocytes in stratum spinosum of the epidermis. As the cells grow out of control, they spread and invade deeper into the dermis as well as up through the surface of the epidermis, typically creating this cratered appearance that we see on the nose of this patient. The damaging effects of UV radiation are major risk contributing to squamous cell carcinoma. These are commonly found on the scalp, ears, and hands, regions of the skin that are often exposed to sunlight. While treatment typically involves surgery to remove the initial malignant tumor, if the squamous cell carcinoma has metastasized and spread to other regions of the body, treatment might require chemotherapy or radiation or immunotherapy, medications that can activate the immune system to help destroy the cancer cells. Here we see the histology of a squamous cell carcinoma, where the mutated keratinocytes are invading into the dermis. Melanoma is a form of skin cancer that forms from uncontrolled growth of melanocytes. Typically, melanoma develops from a melanocytic nevus, although most nevuses are benign tumors. A melanoma is a malignant tumor, and melanoma is the most aggressive and most fatal of all skin cancers because it is highly metastatic and can be difficult to detect before it has a chance to spread to other organs. Usually melanoma appear as asymmetrical brown and black patches with uneven borders and raised surfaces. Treatment typically involves surgical removal of the initial tumor, followed by chemotherapy or immunotherapy. Immunotherapy gets the immune system to attack the cancer cells. Newer forms of chemotherapy called targeted therapies are replacing traditional chemotherapy medications and are far more effective at targeting the cellular signals that drive uncontrolled cell division in the melanoma. With new advances in these immunotherapy and targeted therapies, traditional chemotherapy medications are becoming much less commonly used to treat melanoma. Here we see the histology of a melanoma, where the mutated melanocytes are growing deep down through the dermis and spreading across the surface of the epidermis as well. Warning signs that a melanocytic nevus has become a melanoma can be remembered with the mnemonic device ABCDE. A for asymmetry, because one half of the melanoma may appear different than the other half. B for border, because the border is not smooth, but is instead irregular, scalloped, or poorly defined. C for color, because the spot has varying colors, with one area a different shade of tan, brown, or black, and even areas that are white, red, or blue. D is for diameter, because melanomas are usually larger than 6 millimeters, which is around the size of a pencil eraser. And E is for evolving, because melanomas grow and change rapidly.